Are you tired of biology and want to get back to quantum computers? Well, I want to do that same thing. And the reason for all this biology is to show how the mechanism is actually performed. It's performed by all of this biology. And we can't seem to do it chemically, so studying this biology is pretty important. Now, the title of this thing is Leghemoglobin. Hemoglobin is what gives oxygen to your cells when you breathe in. It grabs the oxygen and it spreads it throughout your body in a nice timed way. And that oxygen release is really important in this case because that nitrogenase enzyme, which is so complicated that we've talked about before, it dies if you give it too much oxygen. But you still need enough oxygen, just a little bit of oxygen, and they call this microaerobic respiration, is that is needed to get the ATP. The ATP is the chemical energy that allows all this to happen. So you need a little bit of oxygen, but not too much, or you're going to kill the whole process. I'm pointing down here at this leghemoglobin, and if we go all the way up, we'll go through these steps very quickly, and hopefully this will be a quick video. Here's what we started with. The plant starts the whole process, not the bacteria. The plant says, with this secretion, I'm signaling the bacteria to come and form this so it'll get down to this point. Well, what's this NOD gene? That NOD factor comes from the bacteria. And between these two things together, then they start this symbiotic relationship. And it starts growing. Now here's the nodule. The nodule is really important, and here's a couple other chemicals that get involved with the nodule. The nodule is where all this happens. If you don't build those ugly looking nodules on the roots of the plant, you're not going to have bacteroids created. Bacteroids are created from their bacterium that came all the way up this way. Those bacterium, once they're in that infection tube, they still start multiplying and multiplying and multiplying, but until we get this nodule formed, and that nodule starts creating bacteroids that are created into the cell. Now these bacteroids have a little insulating membrane around them, so they're kind of part of the cell, but they're insulated from the rest of the cell chemistry into this bacteroid factory for the nitrogen. Then we have some other gene activation that comes from the bacterium that moves us down the road here that starts getting ready for the nitrogen fixing. Now we have the leghemoglobin comes in to the plant, and that plant is really, by providing this leghemoglobin, this is when it all starts activating. The bacteroid can now work with the nitrogenase enzyme and give it just enough oxygen to perform the chemistry, but yet start this whole process going from nitrogen in the atmosphere down to ammonia. And that's the trick. Here's all we're focusing on is this last bar here. If we can do that last bar, then we don't need all those other biology involved. We'll do this synthetically. So that's the challenge.